Good day to everyone. The topic for today is the cerebellum. What we're aiming at understanding today is the function and the structure of the cerebellum. In particular, we will learn how the cerebellum operates and, as a consequence of that, envision its participation in making the animals move smoothly and in the correct sequence. Finally, we will only mention a variety of pathologies associated with misfunction of this organ. As you should remember, the cerebellum is located ventral to the cerebrum and caudal to the brainstem. Several areas with slightly different function have been identified. Here we have the anterior lobe. In the lower frontal area, we find the flcunodular lobe. Caudal to that, we find the posterior lobe. Towards both sides, we encounter the lateral zone. Moving towards the center is the intermediate zone. And in the central part, we find finally the vermis. Basically, the cerebellum receives instruction from both the pyramidal and extrapyramidal system. I hope you remember that the pyramidal system is made of two nervous tracts. One that sends motor instruction to the limbs and torso of the body. This is the corticospinal tract. And the other that sends instructions to the head, face, and neck through the corticobulbar tract. And these carry all voluntary movement, while the extrapyramidal instructions are sent to carry out automatic or unconscious movement that help maintaining posture and equilibrium. In any case, all these instructions are copied to the cerebellum, where they are compared to the feedback instruction collected through the spindles, the organs of vision, the vestibule, as well as multiple other receptors which are monitoring how the original instructions are being carried out. If there are discrepancies in what was intended to happen and what is actually occurring, then the cerebellum is to make the appropriate corrections of the movement as the movement takes place. The details of this will be coming shortly. To be able to figure it out, how does the cerebellum achieve this, we first have to understand the component of the cerebellum which conduct such complex and fast evaluations. The cerebellum, as the cerebrum, and for the same reasons, is divided into cortex or gray matter and medulla or white matter. The cortex of the cerebellum is divided in 3 layers. I have made an artificial separation here because in reality there is no membranes as those that I have drawn in here. Okay? The molecular layer is the outermost layer and contains a variety of structures and cells, such as the dendrite of the Purkinje cells, which are synapsed by the axons of the granular cells, also called parallel cells, because they run in parallel and perpendicular to the distribution of the Purkinje cells. Here we can see the pattern of dendrites of the Purkinje cells in relation to the parallel fibers, crossing and synapsing with them. If we rotate almost 90 degrees, we see the same cell with the corresponding parallel fibers more clearly. The other components of the molecular layer are dendrites of the Golgi cells, which synapse with the axons of parallel fibers, and the axons of the climbing fibers that synapse to the dendrites of the Purkinje cells, and 2 types of interneuron that are stimulated through the parallel fibers and they modulate the Purkinje cells. These are the stellate cells and the basket cells. The next layer towards the inside of the cerebellum is the Purkinje layer. This is the simplest of the layers because it only has the body of the Purkinje cells and some Bergman glial cells. The third layer is the granular layer. It is named because that is the appearance in view that is made of millions of granular cells. This is also contains the Purkinje axons that will connect these cells to the deep cerebellar nuclei. The body of the Golgi cells are also located in the granular layer, as well as the mossy fibers and climbing fiber axons. Finally, we find the white matter, which is called white because it consists mainly of axons, such as those of the mossy fiber and climbing cells. An electron micrograph depicts clearly the molecular layer, followed by a distinct Purkinje layer, and then the granular layer, and in the center we appreciate the white matter. The majority of the activity in the cerebellum is inhibitory in nature. The stellate cells are excited by the axons of the granular cells or parallel fibers, and they inhibit the Purkinje cells. And you can see in the image from where is its name derived. Similarly, the basket cells are stimulated by granular cells, and they also inhibit Purkinje cells. The Golgi cells are stimulated by granule cells, and they modulate the ganglion, which is basically a three-way synapse where mossy fibers coming from different structures ranging from the cerebral cortex, vestibular nerve, spinal cord, reticular formation, bring information which is passed to the granular cells, which are modulated by Golgi cells. The granule cells are excitatory neurons which receive information from mossy fibers, as we just mentioned, and they in turn excite or influence the Purkinje cells. Each Purkinje cell can receive as many as 200,000 synapses from parallel fibers or axons of granule cells. The Purkinje cells, as we have mentioned above as well, they are inhibitory neurons. They are excited by multiple granule cells, climbing fibers, and they are inhibited or regulated directly by the stellate and basket cells and indirectly by Golgi cells. Purkinje cells are the only output for all the processing that take place in the cerebellum, and it sends an inhibitory signal which goes to the deep cerebellar nuclei for further processing. So let's do a very fast review. Here we have the molecular layer, the Purkinje and granular layer, and then the white matter. In the molecular layer are the stellate and basket cells, while in the Purkinje layer are the bodies of the Purkinje cells. In the granular layer, we have the Golgi cells and millions of granule cells. In the white matter, we find the axons of the climbing fiber, as well as those as the mossy fibers. Each Purkinje cell, with their respective basket, stellate, Golgi, and granule cells, constitute a processing unit that processes information, and these units are repeated about 30 million times in the cerebellum, which, if we multiply by about 200,000 granule cells, we end up with about 6 trillion synapses in the cerebellum. We have seen enough of the cells involved in the function of the cerebellum. Now let's try to figure it out, the sequence of the event that leads to the control of the movement. Here we have the brain stem with the midbrain, the pons, and the medulla oblongata. In the medulla oblongata, we find the inferior olive, and in the white matter of the cerebellum, a deep cerebellar nucleus. As you might have figured it out, outside we have the molecular layer, followed by the Purkinje layer and the granule layer. Those are the main elements of this system. The specific cells will be, as you know, the Purkinje cells, the granule cells or parallel fibers, and the basket, stellate, and Golgi cells. Then we have the nerve fibers that come from the premotor area, going to the muscle and to the inferior olive. From the inferior olive, the climbing fibers reach the dendrites of the Purkinje cells and the deep cerebellar nuclei. From different proprioceptors in muscles, the inferior olive receives axons with the information regarding how the intended movement is being executed. Also, mossy fibers bringing information from many sensorial structures connect to the deep cerebellar nuclei, and they also synapse with the granular cells. The last connection are fibers leaving the deep cerebellar nuclei going to the premotor cortex. We start the process with a signal coming from the cerebral motor cortex with the instruction for example, a leg to move certain distance. This information is copied to the inferior olive. The inferior olive then conveys this information through the climbing fibers to the Purkinje cells with a copy of the information to the deep cerebellar nuclei. Through the mossy fibers, a feedback of information regarding how the movement is perceived to be executed comes to the granular cells, again with a copy to the deep cerebellar nuclei. The granular cells then forward this information through their axons or the parallel fibers to the Purkinje, basket, and stellate, as well as to the Golgi cells. This information is modulated by the Golgi cell before it's sent again to the molecular layer. At the same time, both basket and stellate cells modulate the information going into the Purkinje cell. Once the integration is made, the Purkinje cells sends the result to the deep cerebellar nuclei. The Purkinje cells always send an inhibitory signal to the deep cerebellar nucleus. At this point, the deep cerebellar nucleus evaluates the information coming from the Purkinje cell and from the mossy fibers, as well as that coming from proprioceptors to the inferior olives, with the initial request that came from the cerebral motor cortex with the intended movement. If the movement is not being executed exactly as it was intended to be, then the deep cerebellar nucleus sends a corrective signal to the motor cortex in order to trigger a new corrective signal to the leg in order to correct his movement. An important role of the cerebellum is to ensure that the execution of a movement is done correctly. To ensure that this happens, the cerebellum compares the signal sent from the motor cortex to the limb with that reporting back from the limb with the position of that limb as the movement takes place. If discrepancies are identified, the cerebellum then generates the information to correct the movement. A multitude of cells organized in a much prescribed fashion operates in a particular sequence to carry out the comparison and generate the required corrections. The main input to the cerebellum is through climbing fibers, which came from the inferior olives and synapse with the dendrite of Purkinje cells in the molecular layer of the cerebellum. Under the molecular layer is the Purkinje layer, which contains the body of all Purkinje cells and specialized housekeeping astrocytes called Bergmann glia. Under the Purkinje layer is the granular layer, which contains billions of granule cells which project their axons to the molecular layer where they are organized in parallel to each other, forming the parallel fibers, which run perpendicular to the dendrites of the Purkinje cells with which they synapse. Parallel fibers also synapse with dendrites of the interneuron stellate and basket cells. The granular layer also contains the body and axon of the interneuron Golgi cell. In the granular layer, the axon of mossy fibers synapse with the dendrite of granule cells, synapse which is modulated by the Golgi cell, as we can see here, in an unit called cerebellar glomeruli. A very simplified sequence of electrical signals required to achieve the above-mentioned correction is as follows. The motor area of the cerebral cortex sends a signal to the muscle, for example a limb, to execute a movement. This signal is copied to the inferior olive, which then convey this information through climbing fibers to the Purkinje cells with a copy to the deep cerebellar nuclei. At the same time, the inferior olives receives information from the proprioceptors of the muscles, indicating the position and rate of movement of the limb, and this information is passed along to the deep cerebellar nucleus and the granule cells, which synapse with the Purkinje cells. Further, complementary and supportive information from the vestibular system and from the spinal cord are also carried through mossy fibers to the granule cells, which eventually reach the Purkinje cells. All of the above-mentioned connections are stimulatory in nature. The information in the Purkinje cell is modulated directly by the interneuron stellate and by the basket cells, and indirectly by the Golgi cells, which send inhibitory signals. From all of these synapses, which can be as many as 200,000 in a single Purkinje cell, the Purkinje cells analyze this and create the final output, which is a single inhibitory signal generated in the Purkinje cell and sent to the deep cerebellar nucleus. From here, an integrated correction signal is sent to the premotor area of the cortex via the thalamus, if needed. Based on this information, the cortex sends a new signal to the limb to eventually correct the movement as it is taking place. Depending on what activity each area of the cerebellum carries out, this organ is divided into three large sections. The medial area of the cerebellum, including the vermis and the intermedial area, is called the spinocerebellum, and it is responsible for coordinating movement and muscular tone on muscles controlled by signals coming from the pyramidal and extrapyramidal system. So a movement such as lifting a leg over a barrier or jumping a brook would be good examples of the utilization of this area. The lateral area of the cerebellum is also called cerebellum, and its role is to coordinate and to make sure that planned movements are executed properly. Also, sequence of movement that have been memorized are controlled and monitored by this area of the cerebellum. In humans, for example, playing the piano would be a good example of the memorized movement, and walking would be part of the planned movement. Finally, the flocculonodular lobe is called the vestibulocerebellum, which is responsible for the coordination of balance and eye movement. This is the only area in the cerebellum which receives direct input from a sensorial organ. In this case, receives signals from the vestibule. In terms of cerebellar deficiencies or problems, there is a large variety of signs which indicate different types of problems. Without going into the specific detail of each, because it is not the purpose of this course, we're going to only mention some of the most common problems. The white base gait occurs when the balance area of the cerebellum is not working properly. The animal moves with a wide base to compensate for balance problems, to avoid falling out. Ataxia is the name given to movements that are not coordinating, revealing that at least the cerebellum is not working properly. Dysmetria indicate that the feedback to fine-tune a movement is not up to par, because the correction made by the animal are not precise, thus usually overshoots the target. These reveal problems with the spinal cerebellum. A variation of the anterior deficiency is the intention tremor, where the execution of the movement is oscillatory in order to finally get to the proper place. Pass pointing is identified as an exaggerated movement resulting from improper fine-tuning of the movement. If to pass over a 2-inch object the animal lifts his leg 10 inches, that will reveal this type of problems. Dysdiacokinesia is a pathology revealing that the lateral area of the cerebellum is not doing his job. Here the animal executes learned movement, but in the wrong sequence. The cerebellar nystagmus reveals a problem with the vestibulocerebellum because the animal cannot control the movement of the eye and they oscillate in nystagmus. Finally, hypotonia reveals a deficiency in the spinocerebellar area, and it is seen as poor tone in the lower peripheral muscles. In the following clip, we are going to see an animal that has a problem with ataxia. His name is Hogan, and you will see the problems of coordination that this animal manifests. You can see to start that it is very difficult to manage lowering itself through the stairs, and it tends to fall. The animal has a tremendously good temperament, but you can see how awkward is the sequence of movement that he does. They are not very well coordinated. I'm going to let you see a minute or two of this clip. You see the difficulty holding position while he urinates, and the sequencing in the movement of the legs is totally off, therefore it tends to trip and fall. But he's in a very playful mood. He doesn't seem to be affected in his disposition by this deficiency. So his quality of life is not that bad. but I assume that he must get significant bruising from all the banging and falling that the animal undergoes. I will stop it in here and we will continue. In this clip, we're going to look at the eyes of this cat and you will see cerebellar nystagmus. If you look at the eye, they're flicking sideways constantly. It cannot keep them fixed in a point. They oscillate continuously. In this next clip, we're going to see an animal that has a variety of cerebellar problems. We're going to see a little bit of white base. We're going to see ataxia, dysmetria, and pass pointing. You can see that the dog is standing very wide in order to maintain balance. It also oscillates between falling frontwards and backwards, so the whole balance system is compromised. The animal is a lot more comfortable in the carpet than exposing itself to walk in a slippery surface. And probably you saw when the animal moved the legs were very elevated, showing some pass pointing, an exaggerated elevation of the leg to make its steps. Okay? You can see that the steps are very elevated. That's pass pointing, and in here the animal will not get immediately to the piece of food. It will miss getting to it for a couple of times. That is dysmetria. The fine tuning of the approach is not precise enough, so the animal has to try several times. There, the pass pointing is extremely clear with those elevation in the legs. And the lack of coordination revealed the ataxia. And here is going to be a little better approaching the piece of food. But going back, it was a little awkward as well. In summary then, we look at the anatomy and histology of the cerebellum. Then we reviewed the detail of the circuitry involving in generating corrective signals. Then we mentioned the role of the different anatomical areas of the cerebellum in regulating the different aspects controlled by this organ. And finally, we mentioned some common deficiencies. And with this, we have completed another chapter of this course. Have a very nice day.